This is the Part 2 video podcast for Field Methods in Hydrology, Chapter 13 related to water quality measurement. And as you can see, I moved outside just to change things up and get a little bit of a different energy. And what I want to do in this Part 2 video podcast is talk about water quality field methods. Now, of course, a big part of water quality is the chemical analyses that take place in the lab. But there's a lot of things that we can do in the field in terms of what we'd call in situ or direct measurement of chemical attributes of water, in the water as it exists in the natural system. We can also get samples and bring them back. And when we do that, we need to have quality assurance, quality control procedures. And then what are some of the different lab measurements that can be done with that information? So those are the topics that I want to cover. And it's really going to focus on the aspects of the water quality sensors and how they work. It's not going to be so much in terms of the pros and cons of different kinds of sensors, because in this case, across the industry, it tends to be more standardized. If you're going to measure temperature, if you're going to measure pH, the methods are fairly similar. So there's not a lot of different ways to choose from. It's just understanding what all that means and how it works. Temperature is the most basic property related to water quality, and of course a lot of chemical reactions and biological processes are driven by temperature, so we need to know it. And of course it's in units of Celsius, Fahrenheit, or Kelvin, and here's the typical conversion factor. I like Fahrenheit because when you think about it, over the range that we're looking at, you say 1 degree Fahrenheit, like the difference between 69 and 70, something you can feel, whereas it's just not as comfortable to say, okay, well, what's the difference between, 8.15 Celsius and 8.16 Celsius? But, of course, whatever system you're used to is what you're used to, and that's fine. Traditionally, the mercury thermometer was used to make manual measurements of temperature, but now, far and away, the dominant technology is the thermocouple. So let me explain the idea, because the thermocouple is what you're going to get when you buy a temperature sensor that goes with a data logger. So a thermocouple relies on the principle that if you have two wires, such as shown here, like these black polylines are, imagine, wires, and one is made out of one material like nickel chromium, and the other is made at another material like nickel aluminum. And so they're joined at one end, but then if one is at a different temperature than the other one, then that will result in a voltage difference and an electrical current will flow. So the key here is having one as your reference end that you keep out of the natural waters or air or whatever, and one that is exposed to that natural temperature, and then by looking at the difference, a voltage is recorded, and that's what is used to make a calibration with temperature. The main difference in thermocouples has to do with the materials themselves. So if you want to have a wide range of temperature and a high precision of measurement, then you have to go to more exotic metals, and that's what governs that. Okay, the next is pH, which you should all be familiar with from your introduction to chemistry class. It's defined as the negative log of the concentration or activity of the hydrogen ion in the solution. It indicates the availability of hydrogen ions to break down metal salts. So, the lower the pH, the more corrosive it is for metals. The scale is typically 0 to 14, so a change in a pH of 1 unit corresponds with an order of magnitude, factor of 10, change in the concentration of hydrogen ions. So you can't add pH directly if you want to do a mass balance. Like if I have, one solution that's 6 and one is 7, you don't mix them and get 6.5, for example. The pH is temperature dependent. So there are very inexpensive pH sensors that are out there, and there are also very expensive ones. The main difference is really, again, in the electronics and in the quality of the membrane that allows the hydrogen ions to pass through it. Also, consider that with pH, if you have a pH 2 or 3 fluid, you have huge numbers of ions, where if you have a pH of 10, you have like a tiny fraction, right? 1 times 10 to the minus 10 as your activity. So, having different solutions is something you have to be very careful about with using the same sensor. If I take a sensor and put it in a pH 3 and then take it and put it in a pH 10, even if I try to wash it, what's going to happen is you still have so many hydrogen ions present, you're just overwhelming what's the actual condition. But it's pretty rare that you're going to make that extreme of a change. So with these pH sensors like you see in this photo, what happens is you do a calibration on the fly. This could be done, the day before or day of that you do your field measurements. So what you do is, of course, it's measuring a voltage, but what you do is you put it in one solution that may be like a pH 4, and you record it, let it equilibrate, and so now it knows what's the voltage associated with that pH. Now you take it and put it in another solution like a pH 7. So something like 3 orders of magnitude is about as far as you want to go, and of course you want to wash it between each solution, but then let it equilibrate with the second one, and now you have the range over which you can safely measure pH. If you're going to have samples that go outside that range, then I would suggest buying one for like a 2 to 4 pH range, one from 4 to 7, and one from 7 to 10 or whatever, so that you have those. And you can buy the standard solutions, and you'll see that when we play with this in the lab. But this idea about a memory, I'm not talking about the computer. It's like, you've passed ions through into the sensor, so you need to flush those out if you're going to go to a significantly lower concentration solution next. Okay, so let's just think about this in terms of mass balance. Imagine that I have 2 streams, and that's what this weird little illustration is, like 2 streams coming together to form a main stem bigger stream. Imagine that one has a temperature of 15 degrees C, it's carrying 100 cubic meters per second, and it has a pH of 8. Imagine that the second stream has a temperature of 10, it's flowing also at 100 cubic meters per second, and it has a pH of 6. So what are the values on the right side that would be representative of what you would expect in the main stem? Please pause here and calculate those numbers. Okay, well, you can imagine that temperature, well, of course, the discharge is additive. So let's start there. 100 plus 100. By mass conservation, we would expect 200 cubic meters per second. Temperature is a straight linear function, so we can average that and expect that for equal flow to have a temperature of 12.5 degrees Celsius. So pH is the key, and it's not 7. So I'll show you those calculations. First, let's consider the activity of the hydrogen ion, and that's what you have to do here. In one, you have 10 to the minus 6, and in the other, you have 10 to the minus 8. So if I add those together, what you're really getting is 1.01 times 10 to the minus 6, which just helps you to appreciate that a pH of 8 has 2 orders of magnitude less hydrogen ion activity than a pH of 6. So if I take the average of those two numbers, you get 5.05 times 10 to the minus 7. And now I have to remember my formula here, negative log of that number. And if you take the negative log of this number here, you'll find you get a value of essentially 6.3. And notice I'm being very careful about my significant digits. Okay, the next variable I want to show you is electrical conductivity and total dissolved solids. This is a parameter that measures the ability of water to conduct electricity, and it tends to relate to the amount of salts in water. And therefore, it's also sometimes referred to as salinity, electrical conductivity, total dissolved solids. This is an example of 2 different sensors that you can use. A small sensor could be only $30 to $40, whereas this one could be hundreds of dollars. The units are bizarre, but they're micro Siemens per centimeter. And that's pretty hard for a lot of people to wrap their brains around. In contrast, total dissolved solids is more understandable. It's usually in units of parts per million of total dissolved solids. Now, of course, if you don't know what a part per million is, or even if you've heard the term, but do you really understand it? So I'm going to cover that next. Electrical conductivity is also temperature dependent, and it's important to calibrate it with one or more solutions of known conductivity. So you have to buy off the shelf, or you have to very carefully make a solution, from a salt powder or something like that with a very precise mass balance. Okay, so let's just take a moment here and pause, because sometimes it's very common. And when I was a junior or senior in college, I took a lot of chemistry classes, geochemistry and stuff, but it took a while for it to really sink in to understand parts per million and concentration. And a lot hinges on whether in high school you really got a very thorough introduction to fractions and parts per thousand and things like that. So let's start at the very basic level. When we have a set of beads, here's a single sheet with 100 beads. If you count 10 by 10, so we have 100 beads in this sheet. If I consider one bead, that is 1 in 100. So we can call that 1 part out of 100. And we use the symbol percent, which is shown in the upper left, to indicate 1 part per 100. So we say 1%. So 1 part in 100 is something you're already very familiar with. Now, let me take 10 sheets and stack them up to make a cube. Now I have 1,000 beads, and if I look at any one bead, that's 1 per 1,000, 1 part per 1,000. And we use this funky other symbol here, which looks like a percent but adds another zero. We call it a per mil, because mil means part per 1,000, like milli. So we can say 1 per mil or 1 part per 1,000. So relative to those numbers, let's look at our Rubik's Cube. This nice photo here, or graphic, illustrates the idea. Like, of course, when we're talking about a solution, an aqueous solution, you might have something like aluminum or mercury or something in it. Well, it's not like an individual stack of discrete beads. It's more like a mixture, like you see with this melted Rubik's Cube. If you think about a Rubik's Cube, there is an orange. There are orange faces, and there are 9 of them. So out of all the faces, let's see, there's as you can see on the top here, here's like 9 faces, and there's 6 sides. So 9 times 6 is 54. So there are 54 sides, or individual little faces here of orange. Forgetting the black in between, if we were to melt the Rubik's Cube, what would be the concentration of orange that would be in that mixture? And again, considering only melting the faces, not the actual plastic of the cube itself, but just the paint of all the faces. Like if I peeled off all these faces, melted them all together, what would be the concentration of orange? So pause here and work out that calculation for a second. Okay, well, if you think about it, like I said, there's 9 faces of orange out of a total of 54. And I believe if you do 9 divided by 54, I believe you're going to get 0.1666666. So let's just say, so 0.167 would be what? Like, how would we convert that? And if you think about it, a part per 100 would be, would mean that that's 6. If you take 0.167 and you multiply it by 100, you get 16.7. So the Rubik's Cube faces have a concentration of 16.7%, or 16.7 parts per 100. Now let's move on to parts per million. It's the same idea, I just can't find you a nice picture of a cube with a million individual little dots. But it's the same idea. If I say I have 1 part per million, that means there's one tiny little bit out of a million bits. And part per million is a very typical concentration for metals in natural waters. We also have some that might be parts per billion. And for these, we don't have a funky symbol, but we just have part per million. Now, the key about a part per million is everything has to be made out of the same stuff, like mass or volume. Like it has to have the same fair basis to be per. So for example, if I say 1 milligram of stuff, like 1 milligram of aluminum per 1 kilogram of dry mass of dirt, then it's all on a weight basis of grams. But milli is 1,000th of a gram, and kilo is 1,000 times that. So we have basically a difference of 1,000,000 between a milligram and a kilogram. So 1 milligram per kilogram is the same as saying 1 part per million. And since the density of water is 1 kilogram in 1 liter, then we can substitute a liter here for a kilogram, and then we can say that 1 milligram per liter is 1 part per million. So most chemical constituents are reported for natural waters in units of milligrams per liter of water, which is roughly similar to a milligram per kilogram and thus a ppm. Here are some other conversions that you see for electrical conductivity units sometimes, like 1,000 microsiemens per centimeter equals 1 millisiemen per centimeter, and that equals 1 mmHO. So to convert electrical conductivity to total dissolved solids, you multiply by 1.5 microsiemens per centimeter is about equal to 1 milligram per liter. Okay, so that's a little bit of a divergence, but it's really helpful in terms of being able to use, and when you're going out in the field, whether you have a probe that reports an electrical conductivity or total dissolved solids, now you know how to cope with that data. The next variable is one that isn't very commonly used, but I think it's helpful to think about. It's called EH. So you're all familiar with pH, which is related to hydrogen ions, but there's also EH, which is a measure of the amount of free electrons that there are. And this is a measure that relates to the redox potential of an aqueous sample. However, it's highly problematic because EH sensors have built into them many assumptions about the equilibrium state of the sample, and so I found it very difficult to use these. Instead, you can calculate the EH from an equation using dissolved oxygen, and that gets beyond the scope of this course. But if you were to take a course in geochemistry, then you would see that. And EH would be calibrated with a reference solution. Although how that would be done would be tough because it's very hard to, literally keep in a bottle a particular value of EH. And that's true also for dissolved oxygen because, the oxygen, as soon as you open a bottle, it can freely exchange with the air around you. But dissolved oxygen is the amount of gaseous oxygen in the form of O2, like two oxygens coming together as a molecule. And these then get dissolved in an aqueous solution. So you have H2O as your primary medium, and then bobbing around, well, not bobbing, but like bouncing around within that overall milieu in this interstices, there's O2 molecules. Oxygen gets into the water by diffusing from the surrounding air. Also, if you have turbulent mixing, then that can cause aeration, like drops going up or just in hydraulic jumps in a river. And it can also be a waste product of photosynthesis. Because oxygen transfers into water so quickly and leaves water, under biological conditions, but typically if you have a sample that has a really low oxygen concentration from some impaired river or something, and you pull it out and you put it on your table, well, in 20 minutes or an hour later or whatever, it's going to be totally different. So you really have to measure dissolved oxygen in situ or immediately upon taking it out of the water, taking it out of the main water body. And just for some reference values, when it drops below 5, then it's putting aquatic life under stress. And when you get to levels of 1 to 2 milligrams per liter, then that can cause large fish kills. Suspended sediment concentration is the next water quality constituent. We've already talked about it from the case study in part 1 a little bit, but suspended sediment concentration is the total amount of particles in a unit volume of aqueous sample. Now, these particles could be inorganic material or organic material, so that's important to keep in mind. And when we say total amount, it's usually meant by weight, but you could also refer to it by volume or even literally a count of particles, depending on what technology you have to measurement. Usually what you do is you need to collect a sample, pour it into a filtration system, catch all the sediment on a filter paper, and then dry it and weigh it. Now, if you have a pretty big sample or you have very high concentrations, it may be necessary to split the sample, which is pretty undesirable and creates lots of problems. It'd be better to take many small replicates than it is to take splits. But if you have to do a split, then that's called total suspended solids. And so you're only filtering out part of it. Because like when you pour, you're causing a fractionation of particle sizes, and it's nearly impossible to make a perfectly even split. On the right is a device by a company called Sequoia Scientific that can give you the suspended sediment concentration and the grain size distribution all in one shot. One version of it, you put it in the river or lake or whatever. In this version I'm showing you here, you use a hollow tube and like a rubber tube, and you suck water in with a pump. It comes through the machine and then gets dumped out. And so it can be continuously recording particle size distributions and suspended sediment concentrations. So that would be like the gold standard, and I've never had the pleasure of playing with that. So much more commonly, and I should say, that's a very expensive piece of equipment, but so therefore instead, a more common approach is to measure turbidity. Turbidity is a measure of the ability of light to pass through an aqueous sample. Now, it can be influenced by what's dissolved in it. For example, the very first slide, if you go back to the very first page of the video podcast, that lake with that bright blue color, coloration of water itself may not involve particles, but it could be dissolved constituents, and that can affect turbidity. Especially as we commonly see like a tea color if there's a lot of dissolved organic compounds. But predominantly the cause of turbidity, especially as a result of a storm or something like that, is going to be suspended particles. And you can see here a range of NTUs. NTUs are the nephelometric turbidity units that are used here. And you can see going from a very low value of 10 to an extremely high value of 4,000, what that would look like. And indeed, as shown here, you can buy standards and calibrate a sensor using those. So typically what's done is you have a turbidity sensor. It could be a laboratory sensor where you take a water sample and put that in a glass bottle and put it in this little machine that tells you the turbidity. Or it could be a field sensor that you deploy that you actually put in the river, and it can measure the turbidity. If at the same time you take grab samples, then you can establish the relationship between turbidity and suspended sediment concentration, and one of those is shown here, where you could see it's a power function over several orders of magnitude. So like 10 NTU, it's more than 10 milligrams per liter at this site, whereas like 1,000 NTU is under 1,000 milligrams per liter. Okay, well, beyond those, there are many different parameters that can be measured in terms of nitrate, chloride, fluoride, ammonia. And what you need is you need to have what's called an ion selective electrode. What this is some sort of membrane that is designed to only allow passage through of a specific ion. And when that passes through, then a voltmeter is used to measure the activity of just that ion. It's basically measuring whatever's there, but the membrane only allows the thing to be there, the one that you want to measure. And if you look back at the first slide of part 2, let's see if I can go back real quick. In the upper right, these are, I think some of these are turbidity because these have these wipers on them, but you can see a variety, 4, 5 different little, actually six different sensors sticking out here. So you have one data logger. This whole thing could be placed underwater, and then each one of these could be a selective iode, or it could be it could be a turbidity or temperature or whatever kind of sensor. Okay, so this ion selective electrode, that's what I was looking for. So it's making a conversion to a voltage, and then you have a calibration for that. Just want to note that as sort of an aside, but it's an important thing to be aware of. When we talk about nitrate itself, and I think this might come back in the groundwater information, but 45 milligrams per liter is the maximum allowable for drinking water for total nitrate, or if you only report just the nitrogen in it, it's 10 milligrams per liter. Another variable that could be quite useful is chlorophyll. It's bound in living cells of photosynthetic organisms, and it's used as a measure of the concentration of phytoplankton. So that can be an indicator of if there's runaway plant growth somewhere, or in a coastal zone, comparing the relative abundance of phytoplankton. And in order to measure chlorophyll, we use something called a fluorometer. I think I'm going to be coming back to fluorometers in the next presentation related to environmental tracers. But what a fluorometer does is it induces chlorophyll to fluoresce by shining a beam of light of the proper wavelength into the water and then measuring the higher wavelength light which is emitted. So you could do a quick Google search on what fluorescence is and what a fluorometer is. It's a device that we have in the laboratory. There are field fluorometers where you can put it next to the stream and pull in the water and see basically carry that out, or there are laboratory ones. Okay, so switching from specific field sensors, now let's go to sample collection. Because of course there's far more you can do in a lab than you can do in the field, and you can do it in far more detail. So the first thing for sample collection is to answer these questions for yourself. What variables am I going to study? Because then that leads to the next set. It's kind of like a decision tree. Well, I have to choose the type of container. For example, am I going to choose a glass container, a metal container, or a plastic container? And why does that matter? Well, if you have a metal, then choosing a metal container would be pretty bad because it could absorb onto it, or the metal container could contribute materials into the water. An inorganic container would be better there. So I'm sorry, just said that wrong. An organic container. And so organic compounds are composed of hydrogen and carbon, maybe some oxygen, but they're basically designed so that they don't have exposed charged areas that regular metal ions would be able to interact with. So organic containers are good when you want to measure metals, and inorganic containers are good when you want to deal with organic compounds. So it's the complement of things that don't match. Now, coming back to a little bit of the experimental design issue, you have to think about space and time in sampling. When we talk about water quality, it comes down to whether you're just going to get a point, like I'm going to go somewhere and just get the water at that point location and also point in time, or do I want to have some way of averaging, like putting it down through the water column, pulling it back up, and having a little bit of sample come in over time and getting an integrated sample. So there are units called depth integrated samplers that can do that. The same issues come up with sampling in time. Do I want something that's averaged, like have a small opening and let it come in through time, or just open and shut to get that? And when I talk about estuaries and lakes, then we'll look at a variety of samplers. We could do it now, but I'm just going to do it within that other context. There's another kind of device called an ISCO sampler, which is one in which there's a lot of bottles in a machine, and there's a hose pulling water in from the water body, and it puts it in a bottle, and then the bottles rotate, and so it's like you sample on some interval, and a discrete sample is taken out of the water body, put into a bottle, and then it turns. And we'll be looking at one of those on our next field trip. With that kind of sampler, you can decide, do I just want to sample on an interval? Like you could say, okay, well, I'm in the middle of a storm, so maybe it's triggered by a sensor like a water level sensor or a tipping bucket rain gauge or a moisture sensor of some kind. And then I could have more frequent sampling to get shorter term variability, but then you might reduce that to just hourly or daily sampling to get base flows. And if you're looking at groundwater, you might only do weekly, monthly, or seasonal sampling. The most common way of getting a sample is to have a bottle, put the bottle entirely into the water, turn it upside down, right side up to make sure that it's fully immersed and all air bubbles are gone. Take the lid in your other hand. While it's underwater, close it. Unless the water is literally hazardous to your health, in which case you should probably have gloves and appropriate attire. But if it's safe to put your hands in the water, you're not in the middle of a raging flood or you're going to get swept away or something like that, fully immerse the bottle, close it underwater with no air bubbles in it, so you have a 100% pure sample that's not going to undergo mixing with oxygen in the air. For groundwater well sampling or an ISCO sampler or fluorometer, the LIST sampler I was saying for the suspended sediment concentration and particle size, you can use something called a peristaltic pump, and it's illustrated here. So what this does is it pinches a rubber hose, and when it does that and it moves, then that's creating a pressure gradient that sucks the water up. And on the other end, when it opens, then it's literally pushing the water with the second one. So it's a pressure-based pull, and then a direct push to get the water in and out. And so that can then draw the water around. Filtration is the next step. Sometimes you need to filter immediately. That's because you want to separate particles and dissolved constituents right away. If you don't want your particles to further dissolve, then you need to separate them. Or conversely, you may not want precipitation to take place. So filtering helps with that. And so ideally you could split the sample or just collect a sample that you're going to keep as an unfiltered fraction if for some reason you want to know anything about what those changes are. Then we can have what's called a filter apparatus. It's usually some kind of funnel into a flask. That flask is going to have a vacuum, which could be a hand pump, or in a laboratory, many labs have a vacuum line to draw air through. And there's filter paper, and usually that's supported on like a glass porous filter. And so when you pour it into the top, then you're going to make a filtration, keeping particles on top and allowing the solution to move through. It also may be necessary to preserve a sample. That can be done to prevent chemical reactions, precipitation specifically, as well as to reduce the rate of chemical reactions and biological processes. So typically that's done through acidification and cold storage. Quality assurance, quality control has to do with maintaining your level of skepticism and like your expectations of excellence in data acquisition. It's really, really important. I mean, of course, when you're collecting samples, you don't want to go out with dirty samples, right? You need to be very, very thorough. But it can be shocking how very unexpected things can cause contamination. The paper towels or tissues that you use to wipe things clean may themselves be highly contaminated. So it takes a real dedication. Once you have a sample, then you need to check for uncertainty in many different ways. How are you splitting it, acidifying, filtering? There's a lot of different things that you can do on your own. When you take a sample and you send it to a laboratory, because it's usually way cheaper to get chemical analyses done in a laboratory than to maintain all of your own equipment, then you want to be able to test their performance, and there's many ways to do that. One is to send replicates. So if you collect several of the same sample just next to each other, or a few moments apart, then you can send those samples and see, do you get the same result. A split, just basically dividing a sample into two smaller fractions, after you homogenize it by shaking it up or whatever. I don't usually send blanks, but if you're dealing with low concentrated solutions, you certainly can send a blank. And then standards. So it's amazing, but you can buy from the federal government through something known as the National Institute of Standards and Technology, standards of things like, actually there's a San Joaquin dirt sample that you can buy where they've taken that dirt and they have measured everything about it in like 10 different laboratories around the world. So they have a very precise understanding of what its composition is, and they send that to you. And then you could, if you have soil samples you're looking at the chemistry of, you could use that as the basis to compare. You could also just buy from the laboratory individual spikes, like it's just a high concentration of aluminum or manganese or whatever it is. And you should submit those tests to just along with the regular samples. And of course, don't name it blank or spike, like just name it like any other sample, QB1 or whatever, like YR2 or whatever the name, like UCD3. I mean, just whatever it is, so that they can't recognize that's different. And of course, labs themselves are constantly running blanks, spikes, replicates. I mean, they're keeping their line going on a regular basis as part of their procedures internally. And most companies will provide you with their internal QA/QC protocols, which you can then use in your reports. Finally, I just wanted to give you a laundry list of some of the things that you can do in the laboratory as far as analyzing samples that you collect in the field. Many of these we've already talked about, but there are other ones, and it's just simply far beyond our class to go into these. But, carbonate chemistry is a fairly basic and important thing, like alkalinity, of course, nitrate, sulfate, phosphate, hydrogen ion, chloride. Like, these are some of the basics that are there. The major cations in metals, heavy metals that are often of a concern for pollution, nutrients like phosphorus and nitrogen, and then a whole variety of organic constituents. So that's the end of the video podcast related to water quality, just dealing with what are the kinds of things you can measure in the field, what are the different ways of sampling and doing quality assurance, quality control, and then just illustrating the range of lab measurements that are commonly done without actually going into the lab, because that's not part of this course.